Greetings everyone. Welcome to my YouTube channel PathQuest. I am Dr. Gauri Metkar, professor and consultant pathologist. This is the second lecture of lecture series Cell Injury. In this lecture, I will be discussing about mechanisms of cell injury. So let's begin. What are the mechanisms of cell injury? These are the processes which ultimately lead to different types of cell injury. Mechanisms of cell injury include depletion of ATP, mitochondrial damage, influx of intracellular calcium and loss of calcium homeostasis, accumulation of oxygen-derived free radicals leading to oxidative stress, defects in membrane permeability, and damage to DNA and proteins. Let's see different mechanisms of cell injury in details. To start with, first mechanism is depletion of ATP. Various causes of depletion of ATP include reduced supply of oxygen by causes like hypoxia, anemia, ischemia, respiratory and cardiovascular diseases, reduced supply of nutrients like vitamin A, D, vitamin B12, thiamine, and various minerals like iron, calcium, and magnesium, mitochondrial damage by increased oxidative stress, dysregulated calcium homeostasis, and mitochondrial genome instability, exposure of mitochondria to toxins like environmental toxins, drugs, and chemicals. Depletion of ATP can have various effects on the cell, which ultimately result in cell injury. So let's see what are these effects. If we take example of hypoxia or ischemia, which is one of the major causes of depletion of ATP, it results in damage to the mitochondria, resulting in decreased ATP generation. Decreased ATP generation has multiple effects like decreased activity of sodium pump, anaerobic metabolism, failure of calcium pump, detachment of ribosomes, and misfolding of the proteins. Decreased activity of sodium pump has various intracellular effects like increased sodium, calcium, and water inside the cell and decreased potassium in the cytoplasm. Other effect of decreased ATP is anaerobic metabolism, which results in synthesis of the energy by glycogenolysis resulting in decreased glycogen. The byproduct of anaerobic metabolism is lactic acid, which results in acidosis. For optimal functioning of various cells, neutral pH is required. When the pH is decreased, it has multiple harmful effects on the cell, which include decreased activity of cellular enzymes and chromatin clumping. Next effect is failure of the calcium pump, which has again multiple effects which include increased intracellular calcium, activation of cytosolic enzymes causing damaging effects on the cell constituents. Next effect is detachment of the ribosomes from endoplasmic reticulum resulting in decreased protein synthesis and misfolding of the proteins. Misfolding of the proteins results in cell death. So these are the various effects of depletion of ATP. One of the mechanisms of decreased ATP generation is mitochondrial damage. Mitochondria are the powerhouse of cell and they are responsible for generation of ATP by oxidative phosphorylation. So any damage to the mitochondria will result in irreversible type of cell injury and ultimately cell death. Cell death can occur by two mechanisms, that is necrosis, which is disorganized cell death, and apoptosis, which is organized cell death or programmed cell death. Whether the cell death occurs by necrosis or apoptosis in cases of mitochondrial damage is determined by mechanisms which are causing damage to the mitochondria. Mitochondrial damage can occur by two different mechanisms. The one mechanism is hypoxia or decreased oxygen supply or exposure of the mitochondria to toxins or radiations. When the mitochondrial damage occurs by these mechanisms, it results in mitochondrial damage or dysfunction, resulting in decreased ATP generation, synthesis of increased reactive oxygen species, and this ultimately results in multiple cellular abnormalities like damage to the cell membrane and disorganized cell death or necrosis. When the mitochondrial damage occurs by mechanisms like decreased survival signals like in aging, DNA or protein damage, This results in activation of different types of mechanisms. These mechanisms or sequences include increased pro-apoptotic proteins and decreased anti-apoptotic proteins. Apoptotic proteins are the proteins which cause or activate the process of apoptosis or programmed cell death. So when the mitochondrial damage is caused by these mechanisms, it results in increased pro-apoptotic proteins which activate the apoptotic process, and there will be down-regulation of anti-apoptotic proteins. This will result in leakage of mitochondrial proteins and an organized type of cell death or apoptosis. Next mechanism of cell injury is accumulation of intracellular calcium and loss of calcium homeostasis. Accumulation of intracellular calcium occurs by effect of injurious agent on cell membrane resulting in loss of calcium homeostasis. This results in excessive intracellular calcium accumulation and also damage to the mitochondria and endoplasmic reticulum will further release calcium resulting in excessive cytosolic calcium. This has multiple effects which include activation of cellular enzymes. Cellular enzymes are present in inactive forms inside the lysosomes. If they are activated, they have multiple effects on the cell. These enzymes include enzymes like phospholipases, proteases, endonucleases, and ATPase. Phospholipases are the enzymes which cause degradation of phospholipids or breakdowns of phospholipids. Phospholipids are main constituents of the cell membranes. So, breakdown of the phospholipids result in severe membrane damage. Proteases are the enzyme which cause protein degradation, and activation of these enzymes results in disruption of the membrane as well as cytoskeletal proteins resulting in damage to cellular infrastructure. Endonucleases are the enzymes which have effect on nuclear proteins resulting in nuclear damage. ATPases are the enzymes which cause degradation of the ATP resulting in decreased ATP synthesis or degradation of the ATP. Increased cytosolic calcium also gets accumulated in the various cell organelles like mitochondria, resulting in increased mitochondrial permeability, and this will also result in decreased ATP generation. Next mechanism of cell injury is excessive accumulation of free radicals inside the cells. What are free radicals? Free radicals are the chemical species that have single unpaired electron in their outer orbit. Because of this unpaired electron, they are unstable, and because they are unstable, they try in stabilizing themselves, they take electron from the surrounding tissues, and in doing so, they cause damage to the tissue. Different types of free radicals include superoxide, hydrogen peroxide, hydroxyl radical, singlet oxygen, hydroperoxide radical, lipid peroxide radical, nitric oxide, and peroxynitrite. So these are the different types of free radicals. What is the role of antioxidants in neutralizing free radicals? Antioxidants are the species which neutralized free radicals and decrease their damaging effects. So, as we have seen, free radicals are the chemical species which have unpaired electron in their outer orbit and hence they are unstable. So, what antioxidants do is they donate an electron to this unpaired electron, causing the pairing of this and neutralizing these free radicals, and this results in decreased damaging effect of the free radicals. That's why antioxidants play a major role in decreasing the damaging effects of free radicals. So we have seen what are the free radicals and what are antioxidants. So when does a cell is exposed to oxidative stress? A cell is exposed to oxidative stress when there is increase in free radicals and decreased antioxidants. So when the balance between the free radicals and antioxidants is lost, it results in oxidative stress and damage to the cells and cell injury. Let's see what are the different processes which lead to excessive synthesis of free radicals and accumulation of these free radicals in the tissues. So these are the different processes which result in free radical generation. These processes include exposure to UV lights, ionizing radiation, smoking, air pollution and combination of UV lights, inflammatory processes, and various metabolic processes in the body. So all these processes result in synthesis of reactive oxygen species, and this ultimately result in oxidative stress and damage to the DNA. Let's see what are the effects of excessive synthesis of reactive oxygen species. Excessive synthesis of reactive oxygen species results in oxidative stress, which has various pathologic effects like lipid peroxidation resulting in membrane damage, protein modifications resulting in breakdown and misfolding of the proteins, DNA damage resulting in mutations which result in various effects like aging and neoplasia or cancer formation. These are the various effects of free radical injury. These include autoimmune diseases like rheumatoid arthritis, diabetes mellitus, damage to the tissues like liver damage, asthma, cataract, cardiovascular diseases like hypertension, neurodegenerative diseases like Alzheimer's disease and multiple sclerosis, various cancers, and aging. Next mechanism of cell injury is defects in membrane permeability. The causes of defects in membrane permeability include hypoxia, decreased ATP generation, reactive oxygen species accumulation resulting in lipid peroxidation, hypoxia and damage to the mitochondria, and decreased ATP generation resulting in decreased phospholipid recyclation and synthesis. Then increased cytosolic calcium, which results in phospholipase activation, increased phospholipid degradation. All these three processes result in phospholipid loss, which is the main constituent of cytoplasmic membrane, resulting in membrane damage. Increased phospholipid degradation results in accumulation of lipid breakdown products, which again cause damage to the cytoplasmic membrane. Increased cytosolic calcium results in protease activation, resulting in cytoskeletal damage and damage to the membrane. Various consequences of damage to the cell membrane include intracellular accumulation of the calcium resulting in its effects, failure of the membrane ATPase ion pumps which leads to further damage to the cell membrane and mitochondria, damage to the cell membrane resulting in water inflow, cellular swelling, cell rupture and death, and loss of the cytoplasm. Thus, in this lecture, we have covered different mechanisms of cell injury in detail. Thank you for visiting my channel. Please subscribe to my channel so that I will be able to post more and more good quality educational videos related to pathology. Thank you.